Hello, Mr. Otto. So, before we begin, I must insist that you put on these safety goggles. First safety.
Thank you for showing me around, Dr.
Bond. James Bond. No, I'm Will.
So what exactly does this company do, Dr. Eisenberg?
My company is involved in genetic engineering.
And what exactly is genetic engineering?
Genetic engineering is the scientific alteration of the genetic material, DNA, in an organism. Using genetic engineering, we can somewhat control the characteristics of an organism.
Before I invest in anything, I need to know what I'm risking my money over. Explain everything.
You don't mind if I have a seat, do you?
No.
Would you like a seat? Scientifically, the process of genetic engineering is really advanced. In order to alter the traits of an organism, new DNA is first inserted into the genome, which contains all of the hereditary, which is the passing of traits from parents to offspring, information in an organism. Before the process begins, DNA sequences are chosen from two different organisms. The first has the desired trait produced by a specific genetic code. The second is the host organism. There are three important checkpoints in the process of genetic engineering. The first checkpoint takes place at the source of the desired organism's body. There, the DNA strands of interest are duplicated using molecular cloning.
Achoo!
Bless you. Molecular cloning uses restriction enzymes to isolate a specific workable segment of the DNA. A restriction enzyme is one that cuts DNA at a recognized nucleotide sequence. Nucleotides compose DNA. The DNA removed using restriction enzymes is called a sticky end. It easily fits into a molecule called a vector through the exposition of the enzyme DNA ligase. Are you with me so far? Okay. DNA ligase facilitates the joining of DNA strands together in the vector. Once the DNA fragment is connected to the vector, recombinant DNA is formed. This workable segment is known as recombinant DNA because it is a sequence of genetic codes which is artificially combined and would otherwise not exist in a biological organism. The vector is a DNA molecule that will replicate within a living cell. The step of recombinant DNA takes place in a test tube. Next, the vector is placed into a host organism using electroporation. Electroporation is the conduction of electricity to a cell, permeating the outer layer. The vector is easily introduced into the cell, where the DNA does its natural work and replicates as the cell continually divides through meiosis and mitosis. Doesn't it all make sense? Okay, here, let me try to explain it in simpler terms. Imagine that each recipe book is a gene. Each page has its own instructions on how to make a different type of food, similar to how each gene yields its own protein. If genetic engineering were equated to cooking, and if we wanted to make chocolate chip cookies, the process would look like this. Imagine you take out the entire cookbook. One page on the cookbook is taken out, for it holds the exact instructions for how to make chocolate chip cookies. Through copying the recipe, you now have the identical instructions for creating cookies, or DNA from recombinant DNA.
Any business that offers me cookies deserves my money.
You didn't understand the thing I said, did you? Are you with me so far? Okay. DNA ligase facilitates the joining of DNA strands together in the vector. Once the DNA fragment is connected to the vector, recombinant DNA is formed. This workable segment is known as recombinant DNA because it is a sequence of genetic code which is artificially combined and would otherwise not exist in a natural biological organism. The vector is a DNA molecule that will replicate within a living cell. Oh, you didn't understand anything I said, did you?
Yeah, I did.
I'm going to now take you to see Dr. Donazar.
You must be Dr. Otto.
You are?
Dr. Denesser. I am an expert in genetically engineered foods. Follow me.
So what exactly are genetically engineered foods?
GM foods, or GMOs, genetically modified organisms, are crop plants optimized for human or animal consumption using the latest molecular biological concepts.
I'm not sure I understand.
You see, in other words, these plants have been modified in the laboratory to enhance desired traits, for example, resistance to herbicides or improved nutritional content.
Why can't you just do this by breeding plants and not in an expensive laboratory?
You see, conventional plant breeding methods can be very time-consuming and are often not very accurate. Genetic engineering, on the other hand, creates plants with the exact desired traits for both efficiency and accuracy.
Interesting. Can you give me a more specific example?
Here, try this. Plant genetics can isolate a gene responsible for drought tolerance and insert that gene into a different plant making it able to survive through a drought. All plants that come from that one plant will also be drought resistant.
So if you can do this with plants, why have you done this with bacteria or other organisms?
Actually, we have. Genes from non-plant organisms can also be used to genetically alter a crop. The best example of this is the use of Bt genes, a naturally occurring bacterium that produces crystal proteins lethal to insect larvae. These genes have been transferred into corn, enabling the corn to produce its own pesticides against insects.
So why is this worth investing in?
Well, the staggering losses from the insect pest-eating crops are devastating to farmers. Consumers don't want to eat foods treated with pesticides because of potential health hazards. Growing GM food can help eliminate application of pesticides and reduce the cost of bringing the crop to the market.
What about severe weather conditions?
Can we prevent plants from dying because of that?
Yes, there are crops being created that will be able to tolerate severe cold and frost, as well as have the ability to survive through a drought. An anti-freeze gene from fish has been introduced into plants such as tobacco and potatoes. As the world population grows, farmers will need to grow crops in locations previously unsuited for plant cultivation. Creating plants that can withstand long periods of drought or high salt content in soil and groundwater will allow farmers to grow crops in inhospitable places.
So I see how this technology could be useful to farmers, but how does it affect our everyday lives?
Here, try this. Malnutrition is a profound issue in third world countries where impoverished people rely on a single crop, such as rice, as a main staple of their diet. However, rice doesn't contain adequate amounts of all necessary nutrients to prevent malnutrition. Genetically engineered rice to contain additional vitamins will alleviate these nutrient deficiencies. Let me take you to the genetic therapy lab, where you can see more on how this will have a greater impact on everyday people.
Sure.
Follow me.
Play first shot.
Follow me. Hi, I'm Dr. Parker.
Mr. Otto.
Please take a seat.
So what's your job?
I specialize in developing and researching genetic therapy.
Doesn't that involve curing sick people? I think I read about that once.
Yes, it does. Genetic therapy is a process in which DNA is used to treat diseases. It's fairly easy to understand.
How so?
The DNA replaces damaged or dysfunctional cells and tissues with functioning ones, which could cure diseases.
That's great. So I could enter this room with a cold. Will you completely healed?
Well, not exactly.
What do you mean?
Gene therapy is used to treat genetic disorders, disorders you inherit genetically, like cancers, hemophilia, and CGD, chronic granulomatous disorder, and some immune deficiencies.
How simple is this process? It's starting to sound pretty complicated.
Maybe a little. The therapy takes some time. That's also why you wouldn't use it to cure a common cold.
So if I wanted to get a gene therapy treatment, what would I have to do?
Well, there are two very different ways to perform gene therapy. The first takes place inside your body, and the second takes place outside your body.
Which one's more efficient?
They both
have their advantages and disadvantages, but they have the same end result.
So, tell me about the one that takes place inside the body.
Well, in order to transport the functioning genes inside of a cell, scientists employ the use of viruses from entering. In the past, adenovirus was used. This was the virus behind the common cold. Using this method was ineffective because this type of virus sometimes made patients even sicker. Currently, scientists use a type of virus called adeno-associated virus, which doesn't cause a disease in humans. Once inside the body, the virus performs its job by hijacking the cell and forcing it to create copies of its functional genetic material.
Wow, that's really interesting. What's the second method like?
The second method of gene therapy is performed outside of your body. During this process, the blood or bone marrow is removed from the patient and all dysfunctional cells are removed as well. The functioning gene is added to the normal cells and then the two are injected back into the bloodstream of the patient. The cells travel back to the infected area and multiply, utilizing the new gene to create functioning cells and eliminate the dysfunctional ones.
Very interesting. Thank you for your time. I think the most important thing I've learned today is that I should invest in this company, and you should too.
Alright, you don't mind if I sit, do you? Would you like a seat? Sure. Before the process... ¿Tú comprendes que lo...
You can just cut this.